Alright, so welcome for the laboratory portion. Again, we're doing the best we can. Not too much hands-on other than a pencil and piece of paper, I guess. But here's the weekly summary I had mentioned for the class part. I've updated it so you can download. I got the headers right and added the unknown worksheet, which is interpreting the TSI tubes, which I'll go over. Pretty much straightforward. Doing the lab manual and the portions in it, I'll go over that now. From the last time, maybe this handout kind of summarizes what we were doing was identifying and separating different organisms based on some characteristics. And the ones that we focus on that show you different aspects are the tests that work for each of these that are specifically different. And so obviously catalase doesn't seem to work, but the novobiasin and things like that. That's sort of the summary from that portion of the staff. The strep, very same thing, identifying based on hemolysis is usually what a diagnostic lab will use, in addition to the fact that it's catalase negative. But the partial, complete, or no hemolysis, which is alpha, beta, and gamma. Optogen, if it's sensitive, yes, it's Streptococcus pneumoniae. If it has beta or bad hemolysis, or where I call bad, it's where you can see the confluent area zones around where the red blood cells have lysed. And if you look at bacitracin, that separates if it's sensitive, and we've got pyogenes, which that's a bad boy. It causes group A, and we can even test for group A using the sandwich antibody test. The group B, strep agalacti, is negative on sensitivity, so it's resistant. So those are important. In the Enterococcus, we can get no hemolysis, and if we can grow it in the presence of 6.5, but we didn't do that. We're not really that too much concerned. Enterococci evidently has been helping to communicate genetics between some of these really bad ones in the strep species, and also staph too. I'm not really sure. It's kind of new. But that was the deal with last week's lab. Some of us had gotten through it, some of us hadn't in the first round with the staph, and I didn't know who was present or absent during that lab for 2239, but that's okay. BIOS 175 General Micro Atlas. I provided this. What it has inside here are some graphics showing some characteristics of things that are sort of consistent with what we do at the Weight Tech Lab. So you can see the proper streaks. This one is obviously a good one. This is bad because you can see the multiple pass-throughs, where here we have multiple pass-throughs. Is not the way I like to do it. It's not the way I taught you. 1, 1, 1, and 1, and you'll get your single colonies. Macconkey Agar, the actual culture, as we'll see with the E. coli, will turn red. Mannitol Salts, of course, we have the 7.5% salt, and you can see the mannitol breaking down as the presence of the acid. For breaking down mannitol with the dye that's in the plate. Here are the various hemolysis that I was referring to, and there's the S. pneumoniae showing the sensitivity to the novobiasin. We have beta hemolysis, various things that you can see, a mixture of hemolysis with mixed cultures. Zone of inhibition, if you remember when we did the Kirby Bowers, this is sort of a good summary. I would hold on to this for the lab practical. Bacitracin sensitivity, you saw optochin sensitivity with a strep, and novabiacin we talked about already for the staph. Swarming by Proteus vulgaris, pretty stinky organism. Pseudomonas turns green. You're going to actually see bandages if you have a burn patient or someone with open wounds, and the bandage has a green sort of hue to it, then you'll know that it's a Pseudomonas infection. It's very stark. There is a non-pigmented version of it, but typically what I've seen clinically are the ones that turn green. Today we're going to go over TSI, various results, so you can refer to this handout when you get there, as well as the Simmons Citrate and all the indole tests. All those things are in that particular the little atlas that I provided. It's in the laboratory area, and that'll be a good source for study material for the lab practical. Okay, this lab, and this is the handouts that I gave you that are in Word format, and this is dedicated to exercise 12, and gastrointestinal tract, of course. We're kind of shifting to the enteric versions of gram-negative rods. The GNR is gram-negative rod. Members of the Enterobacteriaceae family are gram-negative rods in presence of Peritrichus flagella. In other words, it's bacteria with flagella all the way around Peritrichus. Trich meaning hair, if you recall. And that's highly modal. In other words, they move around. Catalase positive, facultative anaerobes. Sometimes they don't have to be catalase. They can be catalase negative. Facultative anaerobes, and they're oxidase negative, typically. Clinical enteric pathogens can be found with the Vibrio, Campylobacter, Pseudomonas. Those are pretty typical type of gram-negatives that cause clinical disease. Ones you'll hear about are cholera, Pseudomonas Aeruginosa, and that's burn patients, Campylobacter jejuni, which causes a pretty nasty diarrheal disease. Not quite as bad as C. diff or Clostridia difficile, although this would generate a code 9, and code brown. E. coli, ones that I know really well. E. coli, I've studied E. coli for all sorts of things, but it's known for intestinal type, urinary tract infections, and other things. It can cause femur head necrosis in children. It depends. It's an opportunistic, I guess, from that standpoint. It'll grow where it can. Enterobacter aeruginosa. It's a fecal coliform, typical. If we see contamination in water, it's one of the organisms that we look for. Urinary and respiratory is typical. Citrobacter furiendi is found in the environmental habitats, soil, water, and sewage. Intestinal tracts of animals. Furiendi is an opportunistic pathogen responsible for nosocomial respiratory tract and urinary tract. Now, it's opportunistic, and it can cause serious problems. Klebsiella pneumoniae is found in the normal flora of mouth, skin, and intestines. These are nosocomial pathogens, typically causing pneumonia, and also urinary tract infections of the folks that, again, provide opportunity with catheterized, and when they get catheterized, sometimes they get contaminated and place organisms where they normally aren't. Salmonella species. Now, Salmonella is a close cousin or kissing cousin of E. coli, And they're really hard to distinguish Salmonella from E. coli, typically, when we see these. Of course, I know Salmonella species is that's giving a lot of problems in the veterinary side of various things. You'll know if you get food poisoning from Salmonella. It's usually 6 to 8 hours later. Bloody diarrhea, probably best thing to go move on to emergency room. They get invasive pretty quickly, and depending on your immune system, you can have a pretty rough ride. Don't be kissing any reptiles. You can get a good source, turtles and things like that. Salmonella typhimurium, Salmonella enteritidis, most common causes salmonellosis. It's usually gastroenteritis that causes the bloody and invasiveness. Typhi is only found in humans and causative agent typhoid fever. It can lead to meningitis, inflammation of the spleen, fever, diarrhea. The Salmonella typhi is a pretty serious infection. Proteus, it's an inhabitant. It's pretty ubiquitous. It's found everywhere. It's opportunistic, associated with urinary tract infections. Usually not leading to septicemia, but can. It grows very fast. Proteus mirabilis, if you get that on a plate as a contamination, it'll consume the whole plate. Pseudomonas Vulgaris is clinically relevant. It's one that you'll smell at an untreated sewage treatment plant as you drive down. Sometimes they don't have enough money for the deodorizers, and Proteus vulgaris will let you know that they're there. Some non-Enterobacteriaceae species is the Campylobacter. These are the ones that cause diarrhea. We do see it in a lot of poultry, cattle. It's more of a veterinary concern. But there is a foodborne transmission to humans, and infection can cause diarrhea, usually bloody, abdominal pain, and it can cause ulceration, fever, headache, nausea, and vomiting. Pretty bad. So we have Campylobacter jejuni and Campylobacter coli, and those are pretty common. You probably will see those in your practice. Pseudomonas Aeruginosa, it's found in the environment. It's a nosocomial opportunistic. We see it causing UTIs, respiratory, soft tissue infections, burn patients. We see it a lot in burn patients, ones that can't deal with effective immunity when the skin has been so severely damaged. Also, they like to form biofilms, so they stick around, but they have that characteristic green color to it. And there's one other characteristic, is a sicky sweet smell. It's, once you smell it, you'll never forget it. It's nasty. Not as nasty as Clostridium difficile. That will absolutely, it's a tearjerker. Probably the worst on all of them would be Clostridium species. Once you smell one of those, your nose will be damaged for months. It's bad. I'd rather have somebody hit me in the nose than smell that organism. Gram stain, Macconkey, and biochemical tests are good ways to identify enterics. Obviously, you look for the gram negative characteristic, and I want everyone to do a gram stain now. See, we can't do that. A little difficult, and I don't think your parents or your significant others or whatever are going to like you taking BSL-2 organisms. So let's make it a thought experiment and doing a gram stain. And obviously what you want to look for are pink, short, blunt rods. E. coli under our microscope look like equal signs. It's really strange. They kind of line up next to each other, and you'll see just a preponderance of these types of things. E. coli does that pretty nicely. Of course, Macconkey Agar, the colonies will start to turn that reddish pink color that break down the sugars that are in the Macconkey Agar. Then the triple sugar agar tubes are really clever. This requires some thought. I'll go over the Macconkey a little bit better here in a minute. The TSI is a differential test, and this is real important. Remember this part right there. It has lactose, sucrose, and a small amount of glucose. So if I were to stop the video and say, which of the following have smaller amount of glucose by a factor of 10, lactose, sucrose, glucose, or mannitol? Obviously, it would be choice C, glucose. Glucose is the one that's in small amount. The idea about this is some of the microorganisms we test in TSI can't use some of these sugars, and the ones that can use glucose, which is the most common one, we use 10 times less, and it gives us a characteristic when they grow, which one's using the glucose. So we'll show you that. Media also contains ferrous sulfate, an iron sulfate material. When iron gets oxidized, it turns black, and it gets into this hydrogen sulfide. That's the black material from the ferrous sulfate, sulfur containing. So hydrogen sulfide is an oxidation state different from the ferrous sulfate. Phenol red's pH indicator indicates, of course, like everything else, the sugar fermentation. So we have red that turns yellow. In a test useful for the enterics. So what we can do is test a series of enterics in these triple sugar iron agars, and then look at certain characteristics. Now they do have a very similar rollout as how they're read, and I'm going to show you how to do that. But the protocol is you take a TSI slant and you use your loop and you stab it, come out, and then streak up the side. So they show it as you streaking and then you stab, but no, you flame the top after you've prepared your loop and all, gotten your sample. You stab it, bring it out, and then you roll up the side with your loop. It's just one kind of a strike, and then you come up and streak. So that way you're distributing some bacteria in the bottom down here, and then also on the surface of the so there's a stab, a component to it, and a surface streak. That's important. That way we'll properly inoculate the tube, and that is for the triple sugar iron. Now the Simmons Citrate, which we typically see them as green, and it has a bromothiamyl blue indicator, and I'm going to show you the positive negative tests of these. And this one does not require the stabbing part. No stab, just a streak. So you're going to take your loop with sterilely going in using aseptic technique and just rub it on the surface of the slant, and that's it. You don't stab Simmons Citrate. So if I were to stop and say, okay, here's a question: do you stab a Simmons Citrate, or do you stab a triple sugar iron? And you stab a triple sugar iron. Urea agar is a differential media if it produces the urease enzyme. Now urea is of course a component in urine. Urease enzyme hydrolyzes urea to ammonia, the common ammonia smell. The agar slants contain urea and a pH indicator, phenol red. Phenol red is yellow in an acidic environment, and fuchsia, which for those who are not familiar with that particular color, Hello Kitty comes to mind, if that helps. Degradation, you never know that I had a daughter, right? Degradation of urea by urease results in ammonia byproducts, which then increases the pH of the media, turning it into the Hello Kitty color, or that kind of ugly or loud fuchsia color, and I'll show you what that is in a minute. So that's how the technique part of this lab, which we can't do, but we've done this enough in the lab, I believe. Just telling you should just show you just using. Now I already showed you how to go into tubes aseptically, and now this would have agar, and that would show you some of the differences. The rest of these tests are just easy as they can be. We have an indol test, and that means that it's a test for the presence of an enzyme called tryptophanase, and if it produces tryptophanase in the presence of tryptophan, it will produce indol. So tryptophan in the presence of this enzyme will produce indol. And with that, it produces a visible color change, a blue color. Really straightforward. You can see the positive. Down here is a positive blue color, and negative is it's whatever the color the media was, and the light blue color change. And it's a really simple test. It's not all that reliable. Some organisms, they don't produce that much, or the reagent has to be fresh, and I do know that positive color there. Oxidase test, just as easy. So all you need to do is just smear a little bit of bacteria on a filter paper and just dribble, in that case, the indol reagent on top. It just break a little thing and drip it on there. We'll put it on the end of a swab. The oxidase test is run pretty much the same way. This will be a question I'll probably ask, or they always ask on a lab practical. The oxidase test will determine if the organ possesses cytochrome C oxidase, or which is one of the electron carrier proteins for energy, if you recall, in the electron transport chain. Even though many bacteria are capable of cell respiration and not all bacteria have the same cytochrome protein, so we're looking for cytochrome C in this case. E. coli and other Enterobacteriaceae lack that C version of the oxidase, while Pseudomonas Aeruginosa and some of the other organisms do, and that's what we're looking for. So we use tetramethyl phenylphenadiamine, which is a clear reduced electron-rich state, and purple when it's oxidized. In the presence of the organism with cytochrome C oxidase, it produces a particular blue color. It's usually, it's blue. It's a dark blue, but it's still. So the indol positive test, blue, and the oxidase positive test is a dark blue, but still blue, and it's positive. And that's a good way to remember it. You can see it on the handout there. Okay. So is there any question? No question. Okay. Macconkey Agar is a staple in most labs. It's a selective and differential. So if you're asked, so if I were to stop and say, okay, is Macconkey Agar a selective, a differential, or a selective and differential media? And you would say selective and differential. It's selective because it has bile salts, but we're not done yet. What else? It has the same crystal violet as we use in gram stain. So obviously crystal violet's going to plug up all those pores on the outside, or the areas of openness in gram positives, and that just shuts them down. So that's the selective nature. Bile salts is a type of bile that is salty that most of the gram positives cannot handle, and it's a rather vile type of material, but the Enterics can handle it because that's the environment in which they like to live. So the Macconkey has lactose, bile salts, neutral red, crystal violet. So just remember, the bile salts and the crystal violet is what makes the media selective. What makes it differential, its ability to differentiate certain organisms, its ability to break down lactose. What's unique about this one, when it does ferment lactose, the colony turns purple, not the media. And so it's just kind of unique. So deep pink, that I call them kind of fuchsia, bright reddish pink colonies, and lactose or non-fermenters is colorless, is boring, you can see. So lac positive, lac minus, that's what you look for. The citrate test, as I already mentioned, the negative test is sort of a green. So citrate is always this kind of a turquoise greenish color. Positive reaction is blue. So when that happens, obviously the citrate is being broken down as a sole carbon source. Really straightforward. It's useful. We use the citrate test for differentiating fecal type samples. Most fecal coliforms like E. coli are citrate negative, and non-fecal coliforms like Enterobacter are citrate positive. So really clear to see the two there. The triple sugar. Now remember, this was a stab vial, stab and streak. In other words, when you go in with your loop, you're streaking, stabbing. So that's what causes, if it does produce a gas when it breaks down the media, it'll separate the media because you've actually inserted live organisms into the agar. But not, of course, not all of them do that. And so that's what we're testing. So it tells us all sorts of interesting things. Now remember, it has 3 sugars, and they are 10 times, these two, lactose and sucrose, 10 times higher concentration than glucose. So this allows to distinguish between the species that ferment glucose only and those that ferment all 3 sugars. Phenol red is a pH indicator, which is very commonly used. I think we've used it in so many different tests that we've already talked about. Turns yellow in acid and red in neutral, and so that way we can tell if it's sugar ferments. Gas production with fermentation results in cracks or lifting of the media. Iron salts in the media react with any hydrogen sulfide, and it turns this dark black. So to denote these tubes, we have a sort of a protocol. It's an unspoken protocol, but it comes back in the days of the German microbiologists. But if it's a red color, we refer to it as a K. If it is a yellow color, it's A. And so we divide the tubes right down the middle, and so this is the upper and this is the lower portion. So this is KA. You can see the reddish and the yellow down at the bottom. This would be AA. This would be AA with gas. This would be KK and K, and it's got positive for hydrogen sulfide. Now it's sometimes so black it's hard to tell if it's K or not. So earlier you take these tubes out. What I used to do is sneak a peek early in its growth period, somewhere around 8 to 10 hours, and I could tell if the streak, the whole thing was red, or if it had some yellow component before it all turned black. But usually we see it as KK. It's a very rare exception. Does that make sense? This is real important. This is how we distinguish a lot of different microorganisms. And this crappy picture is just identifying some of the characteristics of the acids and the various characteristics so you can get the nomenclature of the K and the AA and that sort of thing. But the harder ones perhaps are the gas and the non-gas, but here's a KA for an example. So you can look at that. Sorry for the quality on that. Urea agar, remember that's the urease activity. Negative is the boring media color, and there is the Hello Kitty color. And so that turns bright pink if it's positive, so that's a positive test. So now we have some good means to identify certain microorganisms. So what I'd like you to do is in your kit there you have a kit that identifies, I thought I had put this out, but I guess I didn't. Identifying the TSI. What you have are some tubes and results, and I want you to match. Just pick one of, there's A through G samples in the TSI actual worksheet part of this, and I filled in one of the examples, I believe it was E, and I gave that example so you could see how it was written, and just choose any of the others, A through G other than E, and just use that as your unknown for the unknown sheet here. So just put the letter A through G and just E, just delete that because I already gave that one. So you can identify the organism and you can score it for the results. I give you virtual results with the tubes and various reactions for each of the letters, so you can fill this out, and then I give you a chart that has the results for the organism based on the various tests, and then you can just identify it here down at the bottom. So this will be one of the things that you can take a picture of and send that to me, so I know that you had done that particular lab right. So it is the add-on to this particular presentation. You'll see the interpretation of TSI is posted that way. So that's the lab. You can go through the terms and various things. If you have any other questions, you can email me, and I think what I might do is to hold a session on Teams if there are questions, or I could do it on Zoom, either way. The Teams are easy because if you can't meet at the same time, that's why I'm not requiring that. Everyone's got their own kind of schedule like I do, and it's really hard to get everyone to meet. So on Teams, if you have questions, you can post them there. I may create a section in there or something called lab questions or something, and you can post them in there. I try to monitor it. For right now, it has been so busy grading and doing all this other stuff that I have to do on the administration side, and then preparing the lectures and with the materials for this, which I don't mind doing at all. I just hope it's effective. It's kind of hard. That's why I have you do the polls for the roll. That helps me know who's around and that sort of thing, just so I can fill in the values on the attendance thing. But it also gives me feedback as to how I'm doing, and speak for improvements. I typically don't teach online. I don't like doing it because I think a science lab is hands-on, and it's really difficult not to be hands-on. However, we had done enough in the lab that we can satisfy it, and the dean had sent around a note for anyone who was questioning about if this is going to impact the evaluation of this particular lab, and it won't. And everything is cleared all the way up through administration, so you're in good shape not to worry about that. So what I would do is just try to head wrap as much as possible and understand what these tests are doing. The actual implementation, we had gone over enough that you know how to inoculate aseptically in a tube, and all we're doing is stabs and streaks and things like that. So I feel like you probably have the technique to do it. I'm not worried about that. But the hard part is the head wrapping or understanding what you're doing, and that's the key piece. So there's enough resources, I think I put in the directory here that you can use. You can talk to each other on Teams if you want. I don't have a problem with that. Share information, whatever you want to do. And collaboration as we get through this thing. We only have a few more weeks. And so we're doing good. Not to worry for you to be upset or anything like that. We'll get through this, and performance will, I'm sure, will be just stupendous, and so we'll get through it. Anyhow, that's it. That's what I have for today. And if you have any questions, just post and let me know. Thank you.